Are we ready? Okay. We're dealing with fat today, lots of fat today. We're going to see how you ingest, digest, absorb, send it around your circulation, send it into cells, and also see how you make fat in your cells, particularly today. Now, I had planned to, during the break, give you some feedback on your team leader talks and your lab books. The system is not synchronising, so I might have to switch over to my computer. Always bring a backup just in case. And also at the end, I'll remind you that your topics for assignment part A will go up on Views this Thursday at 1 o'clock. You'll be able to select them next week in the labs. Okay? Select them during the workshops, but you'll be able to review them at least for a few days before your lab starts. Okay, let's deal with fat, shall we? What's changed in this diagram from the last week? Fat's coming into your gut, into that gut stream, not to the blood, but to the lymphatics. Those fats you absorb, particularly through eating meats and other livestock, for example, and even what we call triacyl glycerols. I'll give you examples of those. I want you to think of fats as long-chain carbons, usually more than 9 or 10 carbons long with an acid function generally on the end. They can vary, we'll see that variation. But those fats change their composition. They're useful for your lipid bilayers. I'll give you examples of that. You see them in the kitchen all the time. There's oil, sunflower, safflower, olive oil, that's a fat. See it in the fridge, butter, margarine, blends of various kinds are fats as well. They're all fats, but you need fats. They're good for you. It's just how much, as always. Too much can kill you. We're going to see these fats come into the lymphatics. They eventually get into the bloodstream, and we're going to see fats come in. I'm going to use the term either fats or fatty acids. Lipids, that's kind of interchangeable. Get used to it. Those fats will come in. We'll see them used to make triacyl glycerols. I'm going to do this a lot to you. I was a star on YouTube or something last year where we used this analogy. A long-chain fat. That's 3 fats. That's a triacyl. The knuckles, 3 of them, is a glycerol moiety. You're going to see them clip off, that one and that one. You're going to absorb that finger and the 3 glycerols in that one into your epithelial cells. You're then going to put them back together. These things make your membranes as well. These are important for you. Can I move to the next slide? Okay. Here is this fat. Look down here in the red box. You see a little kink there, a T-junction there and a kink there. That's a carbon. That's the glycerol. Ring that area there. That's the glycerol moiety. Coming off from that is this OOR, and R means long-chain carbon. It can be any length, and it varies. In you, it's often 16 carbons long. That's what you'll make. But you ingest different lengths, 18, 24, it varies enormously. So again, if you're not paying attention, ring that little section in the middle, that's the glycerol moiety. If I take off this carbon and this carbon, what do I end up with? That system. You're going to do that as we go through today. Now, lipids, as I indicated just a moment ago, do a number of things. What do they do in the kitchen? They coalesce. You have to stop them coalescing if you want to wash the dishes. You use a detergent. The same thing happens in your gut. You use a detergent to break them apart. Because these characters here, you can't cleave off those bonds unless they are separated individually in your gut. They are generally insoluble in water. You've gone to the cafe, you've had a nice little salad, there you are shaking the salad dressing. It's usually oil and something else, isn't it? You shake it up to form an emulsion. We use that in vaccination. You've mixed oil and water. They won't stay mixed with you. You put the oil dressing back down and they separate off. That's what they do. These fats are energy stores. We're going to see breaking them off to make energy. That's more next week. If they have things added to them, particularly here where these carbons are, replacing some of these R functional domains, you might put other molecules on, like phospholipids. If you put a phosphate group on, you make it polar. These long-chain carbons don't like water. With the phosphate on, that does like water. You can change it with other moieties as well, like glucose and like. They can also act as messengers, cofactors, carriers, and the one on the right I want you to circle. I used to make your predecessors go and research these kinds of characters, arachidonates. Have a look at it. It's a long chain carbon, that's what the kinks stand for, a couple of double bonds up there, a couple down there, an acid function. They are key extracellular messengers. They change your metabolism. They change your immune function. Many of the drugs you take, like aspirin, affect them. The thing I want you to write down next to them is they're not stored, unlike other hormones. Adrenaline's stored, cortisol's stored. Many hormones are stored in vesicles and then released like insulin is. But these are made when they're needed, particularly in the immune system. I'll talk about that later if you wish. Along the bottom here, we see this kink with carbons. What's missing off each of those kinks plus the carbon? There's one bond in, another bond in, there's a carbon in the middle. What's missing? Hydrogens, aren't they? We don't write those in, do we? They're missing. There's no double bonds in this character, but that's a fatty acid called palmitate, 16 carbons. If you looked at your will assay for this week, what's it called? If you haven't looked at the will assay this week, you better find out really quickly, because that will assay measures fats. It measures fats. How does it measure fat? It doesn't measure the kinks, doesn't measure how many carbons, doesn't measure the hydrogens. It measures the acid function. You might want to jot that down. That's important for you because this long chain carbon can be any length. As I said, 9, 10, up to 24, 30 odd carbons. It varies. And what you eat will change that variation and change what you do. Changes your brain function as well. Excuse me. These things, unlike glucose, which loves water, some amino acids like water, some don't, these guys don't, so they're hydrophobic. You have to protect yourself. That's why you put them through the lymphatics into vesicles. We're going to see chylomicrons to get them out there. This old diagram I've used for many years I love because it shows you lipids, long-chain carbons, can be used to make triacylglycerol. Write down TAG. You can write that in the exam, TAG if you wish. TAG. If you put a phosphate on, you've made a phospholipid. If you put a sphingosine on, you'll make a sphingolipid, often in immune cells, neuronal cells. Some of these lipids are used to make vitamins. You know, you make vitamin D if you get enough sunshine. Too much vitamin D can be a problem. A, E and K as well. But you also use these long-chain carbons to make steroids. What did I say last week about the adrenal gland? What surrounds the adrenal gland? Not the kidney, the fat. The adrenal gland uses that fat to make steroids like cortisol, aldosterone, and those early sex steroids. If you put a sugar molecule on that fat, you've made a glycolipid. Your immune cells do that. They do that so they can hydrogen bond. If you do advanced cell, I'll show you how they do that so they can stick to other cells and find an infection, traffic to it, and deal with it. But they commonly are fatty acids like this character down here. Now that is key for fats. You need to know these things. Can I move to the next one? And I want you to look at the top for a start, this grey area here. You see this long chain fat? We see the hydrogens, we see the carbons up there. You see a double bond. Now there's journals dedicated just to fat research. Journals, a whole lot of them. You need to know some basics. You see up here we've got carbon all the way through there, double bond all the way to the acid function. If you look down below, you can see there's a numbering system, just like you saw in chemistry 1, 2, whatever you call it. Carbon 1's the acid function. What do you measure in the will assay I've given you? What do you measure? The acid function. So we're going to run along carbon 1, carbon 2, carbon 3. Just note the alpha beta, just circle it. Because we're going to see beta oxidation of these fats next week to generate electrons and protons, i.e. ATP. We skip along and we get to position carbon 9 and number 10. You see a double bond. Now you need to know the nomenclature of these basic fatty acids. This fatty acid happens to be oleic. It is 18 double dot 1 delta 9. So what do you think the 18 stands for? 18 carbons. The double dot to 1? 1 double bond. Whereabouts is that double bond? On the carbon 9 position. You'll get an exam question like that. Tell me what this particular fat is. What's its nomenclature for that? Now, as I said, they can be fairly short. Some get down to as small as 4 carbons, but they are generally 9 to 10, up to around 24. As I said, you make a 16 carbon fat in your liver cells. That's what you commonly make. You have to ingest others, particularly for neuronal cells. They can be saturated and unsaturated. What does unsaturated mean? Is this carbon acid, fatty acid up here, unsaturated or saturated? Unsaturated. It's lost a couple of hydrogens. There's a double bond. Notice there's a kink in it. That's changed its conformation. It's doing something different. If I have a long chain fat with no kink, can they get nice and cosy? Not as much. Now you've got olive oil instead of butter or beef fat. You change the composition and the ratios. It's what you get when you get margarine. You blend in different types of fats and oils. The same general composition, which is what you have down here in this graph. Have a look at it. Down the bottom, you've got olive oil. Does anybody not use olive oil? Down here, you've got butter. I grew up on a farm. We have lots of butter. Now it's more blended margarine. There's beef fat. You go to a butcher, what do you see on that piece of steak? Nice and red. Solid fat until you heat it up, doesn't it? You heat it up, what does it do? It becomes more clear, becomes more solution-like. The ratio of these you see in here, so we'll have a look at olive oil, has carbon 16, carbon 18, saturated, not as much. It has lots of unsaturated, 16 and 18. There it is. That makes it more fluid. Put olive oil in the freezer, see if it freezes. Put nuts in the freezer, see if they freeze. Over here we have butter. We have a lot more saturated fat. You put the butter in the fridge, kind of hard to spread when you first pull it out, isn't it? That's why you use margarine. Stick it in the freezer, good luck. Over here we have beef fat. A lot more saturated. Here still pretty proportional in terms of the unsaturated, but a lot less of the small carbons in there. Because beef make, write it down, triacyl glycerols. They store triacyl glycerols as fat. So do you. In your fat cells, you have triacyl glycerols, these characters. And you're going to clip them off and make them available to muscle cells. Are we comfortable with this one? Can I move to the next one? Is that a yes for everybody? All right, let's ingest fat, shall we? And curiously, do you know if I just gave you fat or oil, you'd go, oh, just a bit, but then, oh, it feels sickly, doesn't it? If I gave you just sugar, a really sweet diet, lots of lollies, you can eat a bit, maybe a bit more, then you kind of feel sick. But if I put fat and sugar together, it's a lot harder to stop eating it. Cakes, lollies, a lot more fat and sugar. It's much harder to tell your intestines, your stomach particularly, to tell your brain, stop eating it. It's less sickly. Let's ingest that fat. So there's my piece of beef steak or my other components that I'm taking in, fish or whatever. There they're going into my mouth. What happens in your mouth with fat? Chewing away? Anything? No? Kind of sticks to your mouth, doesn't it? Often if you're, anyway, interested in high-end food intake, I won't go into the details of that. Particularly if you're in Europe, you'll have alcohol with your meal, won't you? Particularly the French have high fatty meals with often a sip of alcohol of some kind. What do you think the alcohol's doing, apart from their brain and other functions, to the fats in their mouths? Sorry? Yeah, it's a solvent. It's starting to absorb them. It's clearing the palate. That's often what you see. You clear the fats off your palate. I'm going down to my stomach. Here is my fat in my stomach. Has anything happened to that in that stomach? It's just low pH. There's some peptides in there, peptidases. Anything happening with that? No. Mixing it around. Too much fat. Oh, I don't feel so good. maybe a bit of belching or whatever. I pass the stomach, I get to the intestine, so nothing's happened to that fat until all the way till it gets to the intestine. There it is, droplets of it. You have to separate those droplets, so these characters are stuck together. You have to separate them so you can start breaking them up. Let's walk through the schematic. Out comes this stuff, bile. You'll see its structure in a moment. It's based on cholesterol. It's amphipathic. It's hydrophobic one end, hydrophilic the other. It likes water one end, it doesn't like it the other. Your gallbladder produces that. There are many individuals that have gallbladder issues. They have their gallbladder removed, often by keyhole surgery. If you have your gallbladder removed, you have to have supplements or change your diet. There's my gallbladder producing some bile. There's my pancreas also producing lipases and colipases. What do you think a lipase is? It's an enzyme that go after fats. It tells you. That's what the name tells you. ASE, enzyme. Lipase goes after fats. Here they are coming into your intestine. There's the bile. Got 2 triacylglycerols stuck together. What does the bile do? Separates them, surrounds them. Now they're separated. Now the lipase can get to them. What can the lipase do? Clip off that one and clip off that one. This cannot be absorbed through your epithelial cells in your gut. It doesn't go through. It has to be one of those, or that one plus those 3. We kind of clear on that? Okay. So here you are, here's our fat. You're pulling off one fat, one finger, you're pulling off another fat, another finger, and you end up with the glycerol, the alcohol function, the long chain carbon. That you want to circle is 2-monoacylglycerol. Why the 2? Absolutely, the second glycerol. So you clip off that finger and that finger. It stays on the second glycerol moiety. Now you've separated, now you can potentially absorb them into this little character here, Mr. Pac-Man. Write this down, that's your gut epithelial cell. That's the poor cell that only lives for a week or so. It takes in amino acids, it takes in carbohydrates, it takes in salts, and now you see it taking in fat. It's taking in, you see that FA there? Put 2 next to it. 2 times. Why? Because it's 2 fingers coming in. And the 2 monoacylglycerols. Does that make sense? Does that make sense? 1 finger, another finger, 2 of them, and then the glycerol with the fat attached to it. Goes into the cells. It's taken into the cell, into this gut epithelial cell. Now here is where that epithelial cell has to do some serious work. I've just pulled those fingers off. They've gone in separately. Here they are. I now have to put this back together. Why? I don't want that running around on its own. I don't want these running around on their own. So I'm going to make a triacylglycerol in your gut cell. I'm going to put them back together, and I'll show you the chemistry for that in a moment. See that thing there, TG, right underneath it, TAG. It's the same thing, triacylglycerol. You put it back together and you put it in this complex, which is called a chylomicron. I'm going to show you that in a moment. Think about an empty cell, a lipid layer, not a bilayer, an empty cell. It's fats coming together, but those fats are stuck with protein. Proteins ribboned around, keeping them together. The proteins are called apoproteins. It's a whole family of them. So here's my apoprotein, write down protein underneath ApoB. Here are my phospholipids. What does that mean? 2 carbons with a phosphate. Carbons come together to form a ball. The lipid on the inside, the phosphates on the outside. Now I can carry things in that particular vesicle. I'm going to carry stuff from Melbourne to Sydney in a big double D truck. I want you to think of that concept as we go through today. I'm going to send that truck out from my Melbourne epithelial cell onto the highway, full of triacylglycerols and a few other characters we're going to see, things like cholesterol, cholesterol ester. Chylomicrons are big structures. They're carrying fats. Whereabouts are you here? In the lymphatics. That piece of steak you just had for breakfast, lunch, dinner ends up in your lymphatics as fats initially. amino acids we saw last week, into your lymphatics, and eventually gets into your portal system, and the liver starts to work out what to do with all those fats. Now, I had a technician at CSIRO go down to Antarctica on an expedition. Loved doing all that kind of outdoory stuff, climbing. Almost killed me on the second sister, climbing up there once. Went down and was taking seal pup blood. Pull out seal pup blood and looks milky. Why? Because it's full of fat. That's what those seal pups need down there. Your blood can look like that if you have too much fat after a meal. You're now into the blood circulation. The cell has to say, well, here's the double D coming from Melbourne. We have to start offloading whatever that package is, which are triacylglycerols. Well, we often go to a major depot, like Sydney, and then put it on smaller trucks. Let's go and see those. Can I move to the next one? Down here. So this fatty acid, put down times 2. Think of 2 fingers coming together to make that, okay? And I do that deliberately so that you remember it, and you will, if you look at Pinterest or whatever the hell is on there. Now, we mentioned bile salts. Look down the bottom. This is bile. Have you ever vomited up bile at any stage of your life? Really green, yep. Tells you you're really sick, so your stomach's going overtime. The yellow, I want you to note, is the base cholesterol structure. Cholesterol is not a bad guy or girl or villain. Maybe that's a better term. It's required to make steroids because that yellow bit there makes most of your sex steroids and cortisol. Here it is making bile. It's generally a hydrophobic molecule. We make some modifications. We put this alcohol ketone function on the end. What have we made? Hydrophilic end. This is amphipathic. This end likes water. This end doesn't. So it can hang out here but have the hydrophobic bit around the fat. Now, if you don't vomit up bile, generally you resorb most of it through your intestines. Where does it go back to? Through the portal system, back into your liver, and recycle to your gallbladder, back again. That's a good thing because you need lots of it, particularly from a fatty meal. But if you look at your stools, not the one you sit on, the one in the porcelain after you've made a poo, the colour of that, spelt with a FA or FE, doesn't matter which country you're in, is generally because of bile. That greeny brown colour, hopefully more brown than green, is because of bile. Roughly 5% coming out each time. So you need to replace that 5% in your diet as well. So this bile molecule, it's amphipathic, as I talked about, synthesised in the liver, comes from the gallbladder, and it's regulated by this thing. And see this thing hormone called cholecystokinin? Behind that, I want you to put CCK. We're going to see CCK later in the semester when we start talking about hormonal regulation of gut movement, of appetite. CCK, cholecystokinin. And I want you to think of bile as detergent, just like the stuff you use in the sink. If I've got oil droplets in my fry pan, I put detergent, well, you see all the droplets kind of disappear. That's doing the same thing in your intestine, trying to get these characters separated so the lipase can get at them. Otherwise, it can't get at them. Once they're separated, then it can be absorbed. The small point, which is this last bit, carbons 4 to 12 long, we don't need to worry about moving those around with these kind of molecules, with bile. They're too small to worry about. If they're really long, they come together. When they're shorter, it's much easier to separate them. And we'll see that in your blood system later on as well. What they do is they stick to this guy, albumin. Where are you using albumin? PSA. I want you to jot down again, albumin is a common carrier of molecules, particularly extracellular messengers and short-chain carbons. It carries some steroids a little bit. It carries some other hormones a little bit. It's kind of like a taxi, you know, couple in, couple out, off you go, somewhere else, back again, someone else, someone else. Can I move to the next one? Not yet. What I should say and point out a couple of little things on here. You see this thing called a micelle? Not my cell. Remember I said that empty ball, empty cell? One layer of lipids, not a bilayer like a cell. That's termed a micelle. That's also a drug delivery vehicle. You put different things in there, you put it into the patient, you can deliver different molecules, particularly hydrophobic molecules. Or DNA, for example, we talk about an advanced cell. So a micelle is essentially a lipid layer, single layer, that carries things, like chylomicrons. They are micelles. Can I move on now? Okay. Let's have a look at the breakup. Remember, 3 fingers down to the middle one. These carbons have to be clipped off here, carbon 1, carbon 3, triacylglycerol, move one fatty acid, another one, off we go down to this 2-monoacylglycerol. Who clips off those carbons at position 1 and 3? A lipase. A lipase will clip those off. Where do you get the lipase from? Your pancreas. The common one is lipoprotein lipase. But lipoprotein lipase is also stuck on the membranes of your blood vessels. Stop for a moment, think about that. Here's a lipase in your gut clipping that apart. There's my double D truck coming from Melbourne to Sydney, or maybe I'll stop at Wagga or Albury. There I'm going to move just a bit of cargo. What do I need? A lipoprotein lipase to clip a few off. Those cargo positions are receptors on the cell membrane, those lipases that grab them as the truck come by. So that now I can take a few lipids off of that truck before it gets to Melbourne. Sorry, before it gets to Sydney. We're going Melbourne, Sydney this year, aren't we? Yes? Okay. So that way we can start grabbing fats and taking them into cells when we need them through lipoprotein lipase. Write down the acronym LPL. From these chylomicrons. So this big truck doesn't just cart things to one place, it can deliver as it goes. Kind of makes sense, doesn't it? Which means the truck's capacity is being reduced. Now those trucks you see, if it wasn't a double D with the Pantech, the amount of truck you see would decline in size, wouldn't it? I used to drive trucks when I was going through university, put tarps over the top, bring wines up to Brisbane and Sydney. As you offload, the canopy gets smaller and smaller. That's what happens to these chylomicrons. They get smaller and smaller. What happens to these characters? Well, these fats that you eat are often either stored, they're used to make lipid bilayers, like the cytoplasm, the nuclear membrane. They might be used to make other steroids. But the key thing is none of them last forever. They have to be turned over because they oxidise. But the one character that will use them a lot is muscle. Think about where your fat depots are. Generally, where are they? Where work is required. Smooth muscle around your gut. If you're a female, around the mammary glands where you need that energy to produce milk later on. Around your large muscle areas where fat is required for muscle work. Can I move to the next one? Okay. See this kind of fingery looking character here? Just write down that's your epithelial cell. Here it is taking in these fats, the monoacylglycerols, and putting them back together to make the chylomicron. Here they are coming out in this little schematic into your lymphatics. Now you all know your lymphatics are not a pump system, don't you? Very thin-walled, bicuspid, requires muscle contraction. If you have a broken leg, you're in hospital, what do they have to come and do? Physio to get you to move your muscles. If your muscles don't move, if your gut doesn't move, your lymph doesn't pump, it doesn't squeeze all this stuff out. That's why you have smooth muscle cells in your gut to keep on contracting all the time. So in this case, you keep pumping this lymph around through the system back into the circulation. You're moving these trucks from Melbourne to Sydney in the lymphatic system. What you see here is a filled-in version of what's going on. Let's have a look here. The activation of fats. Now, I said last week, and I introduced very deliberately CoA, didn't I? Well, CoA, I kind of hinted at, it's going to be used a lot. Here it is here, going to be used for this fat with some help of ATP to put a phosphate on, activate it, and then bring in CoA. In comes CoA to make fatty acyl-CoA. Which enzyme does the job? A thiokinase. Phosphate's involved. Thio refers to the sulfur in that context. What are you doing? You're activating that fat. Why the hell are you doing that? Well, we're inside this epithelial cell right now. We're trying to put this character back together. What do I have to do? That finger's got to be activated with a CoA to get it back on here. That's what I'm doing here. I'm activating the fat. Here comes my 2-monoacylglycerol, right? 2mg. Here comes my active fat. Look at the board with the laser. That's the active fat. What did I just do? I recycled the CoA. What have I produced? A 1-carbon long-chain fat. There's my 2mg, and there's still one missing. What do I need? Another fat that's already activated. Recycle the CoA. What have I just made? A triacylglycerol in my gut epithelial cells. Does that make sense? What do you do now? You package them up into the double D truck with apoproteins around the outside, and think of the apoprotein as the driver with the delivery note, where I'm going to take this stuff to. That's the information embedded in apoproteins. It says where to go to, what to dock to unload. Some other lipids might be some cholesterol, for example, and a few other characters. So we go via the lymph, then the blood. What if we dumped it straight into your blood? What do you think is going to happen? And think about Melbourne with all these double D trucks coming out of Melbourne, peak hour, full of fat, trying to get onto the Hume Highway. What do you think is going to happen? It's going to choke the highway, just like it's going to do to your blood vasculature. Lymph first, take it slow, get out there so it comes into the circulation slowly. That's kind of important. Can I move to the next one? Yes? Right, let's not block up your arteries just yet. Up here is a chylomicron. See that stick figure? That's this character with one finger back the other way. Kind of like that. Glycerol moiety, one long chain, another long chain fat. There's a whole bunch of them in this thing. You see the cholesterol ester stuck in there as well. Those round balls are the phospholipids. Instead of 3 long chain carbons, one's disappeared, now there's a phosphate group. That's polar, likes water, so it's on the outside. The long chain fats are on the inside. This is a micelle. This is a delivery vehicle. This is that double D truck. That apoprotein, I mentioned there's a whole family. Well, here's the table telling you that. You need to know all of these. Be an easy exam question, wouldn't it? Doesn't help much. But what I'd say is pick out some key ones. Let's have a look. Apo A through Apo B, C, D, and E, they change in molecular weight from around 17 up to about 500,000 molecular weight. That's a protein. But you'll notice HDL, VLDL, LDL. Those acronyms sound familiar? Your HDL, LDL. Here they are up here. There's chylomicrons, magnification, huge. That's just come out of what you had for lunch or dinner. VLDLs are the smaller trucks. I got to Sydney and I put it on a smaller truck. And where is Sydney? The liver. Sydney is your liver. That's going to say what to do with it next. I get to LDLs, we see they're even smaller again, and HDLs. So when you go to the GP to get a HDL, LDL measurement, they don't actually measure anything in an assay, they just look and see how big they are to tell you what your ratio is. That's all they do. If you look at some of these characters here, and I want you to circle, some particularly relate to HDLs, There's chylomicrons, ApoB48. I asked you to see that a minute ago. There's ApoC. What have we got here? ApoC also on chylomicrons, ApoC3 on chylomicrons, and ApoE on chylomicrons. It's the driver with a delivery docket. Sometimes the driver stays with that same docket. So I'm kind of clear. Now my double D, I unhitch the back trailer, unuse the front trailer. That's how you work. You unload this payload. If you look at chylomicrons, you can see the proportions along the x-axis. We've got TAGs, protein, cholesterol, cholesterol ester, phospholipid. What have you got there? TGs, triacylglycerol. That's your load. That's what you've had in your meal. That's been put into that chylomicron. So as I drive from Sydney to Melbourne, I get to Albury, I offload some of those. I get down there now to, where are we going to be? Goulburn. We're down to, say, roughly 50%. Just doing the drive. What happens? The ratios of these change, don't they? There's less triacylglycerol. My truck's more empty now. I can go faster and get a speeding ticket. But I'm delivering fat as I go. I'm delivering it to those LPLs, to other components as I go around your body in the blood circulation.
So they get dropped off before they've gone to the liver?
Some do, okay? And the reason I'm trying to harp on this is because this chylomicron starts off as a double D in Melbourne, but as you go, it gets smaller. Albury smaller, get to Sydney smaller. Yes, I might do some other things, but reality is the truck's load just gets smaller. Move the trailer, gets smaller again. The proportions change. And that's all you see when you go from chylomicrons. The key bit I need to alert to you is chylomicrons are made where? Where are they made? Not in Melbourne. In the gut epithelial cell, okay? One you're going to see in a moment, the VLDLs are made in your liver by the fat you make. The chylomicrons and these characters carry around what you ingested for lunch, dinner or tea. Supper or breakfast, whatever you have. Is everyone comfortable with that? Can I move to the next one? Okay. And here's a nice little schematic showing you just that. Let's have a look at the schematic. Incoming chylomicrons. There's my double D. I eventually get to our blood vasculature. What have I got around here? Circle, lipoprotein lipase. Put in the LPL. What are those characters? Stuck in the membrane of your blood epithelia, grabbing fats as the trucks go by. Not just in Albury or Goulburn. It's like someone on the side of the road. Give me a fat. Give me another fat. Pull over. And as you go, what happens? Your truck gets smaller and smaller, and you move them around. They get back to the liver. The liver says, okay, great, we'll manage them. What comes out of the liver? What's coming out of the liver? VLDLs. So VLDLs, please put next to that, that's the fats you make. And you make a carbon-16 fat, normally with no double bonds. If you look at the table, should we memorise tables? I'll tell you when tables are really important to know, absolutely. This table, look at a key example. Let's wander through it. Chylomicrons, their density, usually less than 1, not much protein, phospholipids relatively low, cholesterol ester relatively low, free cholesterol ester relatively low, TAGs, just what we saw in that graph, isn't it? Run down through VLDLs, LDLs, HDL, the proportions change. What happens to the amount of triacylglycerols you're moving around? A lot, isn't it? Yeah. What increases, particularly phospholipids? Why? Because the truck shell, its body, its tray, the cab, the wheels haven't changed. Still the driver in its delivery docket. He hasn't lost any weight, or she, whoever they are. Could be a robot now. So that gives you an idea of what changes the proportions. And again, your GP just measures the size to tell you what your HDL-LDL ratio is. They're not going to do that will assay you're going to do next week. And what does that will assay measure? The free acid function. It's going to come and bite you very soon. Can I move to the next one? Right. Now I have a little animation to show you for a change. Remember I said these LPL proteins, these enzymes sitting on the surface of the cell? Well, here's a classic cell. Get rid of that garbage. Here's a low-density lipoprotein. Those blue balls are the phosphate group. It's filled with fats. There's our apoprotein, that blue ribbon running around. Down here, you're seeing the lipid bilayer of a cell. That's the same thing you see in the still version over on the right. Here is our truck trying to go from Melbourne to Sydney, and someone's on the side of the road. Hey, stop, pull over, give me some fat. Here they are on the side of the road. There they are stuck in the membrane. What are those? LDL receptors. Low-density lipoprotein receptors. They are the docking station where the truck's going to back up to, forklift, get all the fats off. What you're seeing a little bit of here is some advanced cell where we're going to use this thing called clathrin to make a vesicle. Let's make the vesicle. Clathrin forms a little cage. We form a bubble with that particular lipid bilayer from the cell. We take in that vesicle. Hang on a sec, didn't I just say the truck backs up, we offload and the truck keeps going? Not always. Chylomicrons do because they're big. But the smaller the truck, the more likely the cell can ingest it. This is like pinocytosis. We saw that last week, didn't we, with antibodies. Your gut cell has to pinocytose antibodies from the mother when you're first born. Here we have the truck being engulfed by that particular docking station. We're not just pulling out fats now, we're going to take the whole truck. We're going to recycle everything. Driver's going to work for me. Cart note I'm going to use the wheels on another truck and all the rest of it. I'm going to recycle everything in this vesicle. And the fats are mine. How am I going to break that up? Well, your cells break it up, ready to recycle everything. Recycle the clathrin for another ingestion and absorption. I need someone to get in there and break up that skeleton, break up the truck, pull out the fats. What do I need? Some acid. You've got a particular pump, just like your gut pumping protons in there. You might well recycle the receptors for that truck. Awfully slow recycling, isn't it? I'm going to break this thing apart now. There we are. I'm going to hand it off to a lysosome. Separate every component. Some of those fats are going to make lipid bilayers for the cell. If it's in a steroid-producing cell, if it's in the gallbladder, could be there to help make some of those. The bits of the truck are pulled apart. The cholesterol ester are used for other things within that particular cell type. You've just recycled the whole thing, not just the fats in there. But remember, the chylomicron is the big vehicle, moving lots of fats around. In the fed state, we see over here, chylomicrons in, they move around. This thing here, this lipoprotein lipase, is an enzyme stuck on the membrane, just like someone on the side of the road. Give me a fat. Give me a fat. I don't want to stop the truck. I don't want to recycle the truck. Just give me a fat. And I keep moving around until the truck gets more and more of its load reduced. And who's going to use that fat? Well, muscle. Side of the road at Goulburn. I need to work, I need some fat for energy, not enough glucose. And your dear old fat cells say, well, there's lots here, I'll have to put it in storage, that's my job. And I want you to write down something rather traumatic for fat. These fat cells get bigger. You'll see how they release fat in a little while. They get bigger like they do in you. You're born with a number of fat cells. Until they get so big, they're told to divide. That's when the trouble starts, because now you have double the number of fat cells. What do? Get bigger. The trouble is now you've gone over what you started with, the number of fat cells you were basically nominated with, to doubling it, which becomes an issue for you long term, doesn't it? How much you can store. Can I move to the next one? Getting worried about fat now? Some is good. Too much is not so good. What kinds of fats do we have? Well, there's our glycerol, carbon 1, 2, and 3. Here's our long-chain fat. Here's one with a kink. Now, your phospholipids, the ones that are in your membranes, often have the kink carbon, long-chain fat, on position 2. Jot it down. In glycerol, carbon 2 often has the double bond fat that's kinked. Might be one double bond, two, might be a few extra. That means that middle finger has a little kink to it, which means when I put it next to another one, it doesn't want to get stuck. It's not like butter now, it's more fluid. That's what your lipid bilayers should be. I'll show you that shortly. That's where we see the double bond commonly. Write down the word commonly. It can vary slightly and depends on your diet. You can change that. My colleagues in CSIRO were feeding fish meal and fish oil to lamb. Why would they do that? Well, Japanese were buying lamb, but they don't like the smell of it generally while it's cooking. So you can change the smell of fat by changing what the sheep eats. Change its composition. I tasted it, not my cup of tea, but it doesn't smell like lamb when it's cooking for the Japanese. So it's a huge market in Japan and Asia by changing what those lambs eat, to change what's going into their fat cells, to change how they smell when they're cooked. These molecules make up the lipid bilayers of your cells. But your cells make other things. I'll go into detail next semester. What you have here is fatty acid elongation. Where does that occur? In the mitochondria. You're going to see that today a bit. Down here in the ER, you know what rough ER is, don't you? Yeah, what is it? What is it? Yeah, I got that bit, but what is it? It's got ribosomes on it. It's making polypeptides. Busy doing lots of work. Tells you what the cell's doing, because if there's lots of smooth ER, it's not making many polypeptides. But smooth ER is where you often make lots of fats, particularly in some cells. These fats have different classes. So I mentioned before the storage fat, what that bear stores, what the cow stores, what the sheep stores, are mostly triacylglycerols. That's what you store. Triacylglycerols. How many carbons do you store in your triacylglycerols? How long is the carbon length? 16. That's what you can make, and I'll show you why shortly. Linked to the glycerol. Some of these have polar head groups. Here's the phosphate with an alcohol. Here's another phosphate with a choline. Glycerol, phospholipids and sphingolipids particularly. They are in lipid bilayers. Membrane, nuclear, cytoplasmic, ER, you name it, mitochondria. Mitochondria varies the inner membrane a lot to keep protons out. These glycolipids, write down mostly immune cells because they put the carbohydrates. You know your ABO blood group is a function of the carbohydrates on these fats. That's what gives you A, B and O. I'm not going to so you know blood group A, blood group B, blood group O, yep. That's a function of the carbohydrates on there. Now when you think of carbohydrates on a molecule, think of lots of water but hydrogen bonding. They'll stick to something. And that's what immune cells do. When they put carbohydrates on the cell surface, they stick to epithelial cells. They stick to somebody to do their work. It's a way to tell them what to do. And if you have lots of carbohydrates, I'll talk about later on, in your diet, you put lots of carbohydrates on your immune cells. And what do you think is likely to happen longer term? Have a guess. If your immune cells are loaded with carbohydrates on their surface, what do you think they're going to do? A lot of things. They often cause autoimmune diseases. Too many carbohydrates load up those immune cells. They start going after you instead of doing their job protecting you, which I'll talk a bit about after the break as well. That is mid-semester break. We done with this one? Okay. Just a couple of simple examples. You know, for me, this is kind of first year stuff, but up here we see the blue. Think of phospho groups on the ends. Those yellow kinky bits are actually, not kinky as in kinked, but as in carbon, hydrogen. Those are saturated fats. Over here you have some unsaturated. What does that do? It changes from beef fat, solid, to olive oil, more liquid. The physical properties change of these fats. That's what your mitochondrial membrane does, the inner membrane. It's less fluid, doesn't allow protons through. The glycolipid, here's a long-chain fat. That's what the R stands for. What have you done? Stuck a glucose or a galactose. It could be lots of different sugars I'll talk about next semester. Cholesterol, there it is there. You know the base structure. Down the bottom, you see a lipid bilayer. Those little pink things there are the polar head groups, the phosphate. The green bits are the long-chain carbons. Those blue things running through, I want you to jot down, are integral membrane proteins. Those characters do all sorts of things. They could be receptors, they could be pumps, like a glucose transporter we're going to see in a while. They could do all sorts of things to cells. What I'm going to show you now as I show the advanced cell students, I'm going to turn the lights out so you can have a little snooze. This is a live cell. That's the edge of it. There's a long filopodia, and that's the other edge of it, stuck on a dish. You're about to see some laser tweezers come down and grab the lipid bilayer and move around. And as it does, I want you to think about an oil droplet in the sink. No detergent. There's my oil droplet in the sink. There's some water. My oil droplet's kind of moving around, looks really good. Stick my finger in it, and I can get it to do all sorts of things, can't I? This will give you an idea. Now, this is a live cell. This is in real time, showing you how mobile this lipid bilayer of your cell membranes, the cytoplasmic membranes, is. I'm pulling up the lipid bilayer. Have I created big pores to let things through in? No. I've just moved the droplet. Have I changed the cytoskeleton of the cell or any of these integral membrane proteins? Some of them wander around like kind of ships on the ocean. Some might be anchored, don't move, but their oil whizzes past it. This tells you how fluid your lipid bilayer is. And immune cells have to do that when they're trafficking around your system looking for bad guys or girls. You can see that's microns, we're stretching it. The cell's around 20 to 30 microns across. That's a couple of microns. I'm stretching a lipid droplet, and I'm not breaking the cell. I'm not putting a hole in that cell membrane. Thank you, Stephen. And over on the right, what have you got? A micelle. What are you going to put underneath that? Chylomicron, VLDL, HDL, LDL. What's in there? Fats. What I want you to also write in there, drugs. Hydrophobic drugs are put in these micelles because this thing confused with the lipid bilayer of a cell. Many drugs are delivered via micelles, and you can make them very complex. So this lipid composition is critical for you and your cells. You've just ingested some to make more cells. So you do need fat. We done with this one? Quiet. We're not going to go into any more cell biology at this point. That's for next semester, if you're brave or silly enough to do it. Done with this one? Okay. Maybe it's time for a break before we have fatty acid synthesis. Okay. I'll give you till 5, and then I'll give you some feedback on your lab books and team leaders. Okay.
To have our aim and method on the slide, because I had it in my actual presentation, but I got marked down because it wasn't on the slide. I'll copy that.
Let's give you a bit of feedback on your team leader and your lab books. Chris doesn't want anything. Don't need to know. Now, I went through the alanine assay in week 1. I told you this is a standard curve. It had an axis and it needed numbers and a label. Down here, the x-axis had numbers and it needed a label and a unit of measure. Now, I've seen quite a number because I looked at the assignment Dropbox. I saw lots of different forms of the team leader presentation. Doesn't it say summarise your work? Pictures of plates do what? If I looked at this graph, I had one, but unfortunately that system is not working correctly at the moment, and I have nothing there, and nothing there, and nothing there. What would that graph tell me? Nothing. That's the problem. That's why you use that will assay as a guide or a template. It has a title, doesn't it? Doesn't it summarise a bit more than a page, but you can do that in a sentence or two of what's going on. Your team, what beverage did they use? Or should I say plural, beverages did they use? Did you summarise their data in a table? And I've seen some team leader PowerPoint slides that are so busy I can't work out what the hell's going on. And I see hand-drawn stuff. Now, this is a presentation. We're trying to get you professionally prepared so that you can get the job and keep it. If there's no units of measure, you won't be keeping your job. If it doesn't have a title, you won't get the job. Those things need to be on there. That's why they're on the marking rubric. We asked you to give us an equation of a line so we know what it is you're doing. You need to summarise what your beverages do. This week you're using an animal-based beverage, aren't you? Yes? Milk, fish oil, oyster sauce, I don't care what it is. It needs a nutrition label. Why do we ask for a nutrition label? Just for the hell of it? Why would we ask for a nutrition label? He's not well person. Why would we ask for a nutrition label? Because it tells you roughly a protein concentration. Isn't that plant-based beverage based on seasonal activity? I hinted during the lectures there might be seasonal effects, less protein because there's less protein available for the plants to manufacture, less sunlight, less CO2 or more CO2, whatever the case is, less water. It varies. Those characters that measure that do the same assay you're doing. Are they doing the same assay you're doing? Go like that. They are. Yes, they are. So your assay should measure it. Do you use the beverage as a standard? Why would you do that? That's the unknown, isn't it? You've got an estimate, and you had an estimate, so why didn't you look at the concentration and try and get it onto your standard curve? Didn't think that far ahead? That's what problem solvers do. Look at the nutrition label. It tells you roughly a protein concentration. Mgs per mil, whatever it is, what's your standard curve run from? What was the minimum, what's the maximum last week? We gave, sorry? What? What's the units of measure? Micrograms or milligrams? You need to know which one it is. Because this week, what are you doing? What's the focus of assays this week? Sensitivity. How low can you go? What's the lowest concentration you can detect with what? Your milk sample or BSA? Which one? BSA, because we've told you that's the concentration. Last week we gave you a concentration, a nice, easy, straightforward one. This week, don't be prepared for an easy, straightforward one. It's got a different unit of measure, a different concentration, but you know what it is. We've made it up to be exactly that. Or I should say the technicians have. That's your known. That's the standard along here. Not the beverage. Should I plot my amino acids along there? No. Did you get some blue from those amino acids? But did you ask yourself what concentration you detected those amino acids at? It was all on the labels. Were they there in your lab books? Probably not. All those things are missing. You look at the rubric, it's the same as the will assay. If that will assay doesn't have those basic information sets, is the will assay worth anything? Totally useless, which is kind of like your lab book, isn't it? If you give that lab book to somebody else, and that's why we say use a professional one, because that lab book has to go to a patent court where you might be defending it. If it's missing a unit of measure, missing some detail, it's totally useless. Your company's just lost that patent litigation. You've lost your job because you haven't done the really basic things. Now, some of you did a lot of work, put all the amino acids in, did all that stuff, and you just missed units of measure, which means a lot of effort went into a nice round number, didn't it? That's what you have to do. Now, we're not assessing whether you make mistakes. I told you that before. Some of you didn't dilute, so what do you do next time? Dilute more. Some of you didn't look at the wells. Was it mixed properly? Were there bubbles? Were there different volumes? I made all those hints. Didn't pay attention? It affects your assay, doesn't it? Did you plot a standard curve? Some of them look like that, some look like that. That's not a big deal because you're going to do it again and again and again till you get it right. Because when you finish this unit, you need to get it right, otherwise you won't get a job. And these are not difficult things, they're problem solving. Look at what's going on. We asked you then to do the will assay, didn't we, in your team? That's essentially what you do in the real world. I've got this problem. I don't have a lecturer or supervisor tell me exactly what to do. I have a group of people who have sort of an idea, but I have all these resources, my web page, my Excel sheet. I work through calculations. I can use examples. I can go to the web. That's your problem solving. That's the hour you have to do that. So do it. Now, those results or answers will go up on Views later this week, about 1 o'clock Thursday, or soon thereafter, so you can see what you should have got for that urea assay. And those assays are straightforward because everything's telling you what to do in those assays. Except what I do is tell you there's a dilution factor of some kind, don't I? You have to account for that, do some other extra step. Can you find that out? Of course you can. Khan Academy or something online to help you. That's what you do in the real world to solve the problem. We're not going to be in there giving you little answers all the time. It's your job to sort that out. Fortunately, it's only like bugger all marks. But that's your job, problem solving as graduates. That's what your employers want. That's what the federal government wants. How you sort those things out. This is the industry standard. It's not my university one fill in a box because I'm all happy because you got the marks I wanted, or you copied or colluded with someone. This is what you need to do. And if we gave you lab manuals like your lab books were currently to first years, they'd be screaming at you, wouldn't they? It makes no sense. I can't tell what you're doing. But you just imagine the poor markers, those demonstrators trying to mark your books. I don't know what you're doing. There's a guide, a straightforward series of steps. Each will assay. Last week it was the urea. This week it's the lipid one. I've asked the supervisors to tell you what's the similarity, what's the difference between each will assay. Well, I showed you the alanine in week 1, didn't I? You went through the urea assay the last couple of weeks. You're doing the lipid assay now. What does the lipid assay measure? The acid function. Why? Because the lipids can be any length, can't they? Every double bonds, more or less double bonds. I'm going to measure one end of it. So now think about that for a moment. If I've got a fat that's only 9 carbons long with all its hydrogens and one that's 36 carbons long, has it got a different molecular weight? Of course it does. So I've got to account for that. I'm just measuring the acid function. That's what these assays do. They vary what probe they use. Look at that for information. Now we'll give you more feedback after the team leaders for next week, but Those of you who've been team leaders already, take a sigh of relief, because like we tend to do is you've got now the benefit of feedback, don't you? But you've just done your talks. You're going to be given the opportunity to redo your team leader role in Module 4. All is not lost. But your lab books, that's another story. They have to keep on improving. So team leaders, take note. If you were here last week giving your talks, if you were away pretending to be sick or otherwise, doesn't matter, you've got to do it again. But those of you who've done the job of team leader, you've got a second bite at the cherry. It'll be optional, totally up to you. Is everyone clear on that? Why? Because everybody else gets the benefit of feedback, don't they? Just totally appropriate, isn't it? And we can't have practice on practice because we need to get you into it. There's not much semester, the university doesn't give us much money. So this is what you need to worry about. Keep looking at those will assays. Look at their instructions. Look how they write. Do they write whole paragraphs of kind of garbage bump? No, they get to the point. That's what you need to write like. As graduates, most definitely. Now, I'm going to summarise all that information, including the stuff the supervisors and demonstrators said. C1, V1, couldn't do it. Couldn't do an equation of a line. Didn't look at the wells. Didn't look at the mixing. All those things are going to be summarised on a document I'll put up on Views later Thursday. If you're not paying attention to them, NMP. It's not my problem because we're giving you the feedback. Look at the marking rubric. It asks you for a title and a name. Not my title and name, what you did. What beverage did you use? Which amino acids did you use? Is it in your title, in your aim? If it's not there, it's not accurate, is it? Simple as that. It's either there or it's not there. If it's there, it's 1. If it's not there, 0. Got that? Just got to put it there. That's all you have to do. All right, shall we get back to work? That was scary, wasn't it? Sorry, when you get the marks, what was the question? Good point. This week, when you come into the lab, you'll get your marking sheet back. It'll have some comments on there. If you're a team leader, it'll also get your team leader comments back as well. So you'll know what you did wrong. The will assay, the actual answers for those, will be up on Views. Now, a couple of small things before I get on with this stuff. A couple of people asked me about the assignment Part A topic that'll be available next week in the workshop. You'll get to select them in the workshop. Is that kind of clear? They'll be available from Thursday at 1 o'clock, as I said at the beginning of today, for you to have a look at, to decide, pick some priorities, because you may not be the first. The supervisors will decide how you get to go first. So you may not get your first choice. It's a limited number of topics, 40 maximum. You get to pick one, but make sure you collect a few that you're going to have as options. Next week. Okay. Let's start with fat synthesis. Why are we doing this? Because last week we pulled the nitrogen off and we had excess carbons, didn't we? And some of those excess carbons can be used to make fat. They can be used to make glucose more primarily, but here we're going to see the process of making fat. You can also make fat from what? What is this particular image telling you right at the top left? Glucose. Lots of sugar, you turn it into fat. I keep telling you, humans are particularly good at that. Glucose running down through glycolysis, lots of steps. You get down to the bottom to pyruvate. What do you make? Pyruvate's got to go into this, looks like a cross-section of an orange, doesn't it? What do you think it is? You are an hepatocyte, yes. That's this whole thing here. But that round thing that looks like a cross-section of an orange, what is it? Starts with an M. Mitochondria. Shake your head. You have to yell out, I can't hear you, I'm old and frail. Whereabouts are you in the mitochondria? Inside that inner membrane space or the matrix? Matrix, what's happening in there? Your old friend OAA's back again, isn't he? Or she? This thing, acetyl-CoA, what's that? CoA, the carrier, and 2 carbons stuck on it. Yep. What do you do with that? You make citrate. Didn't we see a bit of citrate work last week, running around the urea cycle, some other stuff going on? Here's citrate coming out. So excess glucose, what did my liver do? Fill up glycogen first, just note that. Fills up glycogen first, keeps the blood happy with glucose, but if all those are happy, what do? Turn sugar into fat. And any excess carbon I'm going to turn into fat. I run it through glycolysis, keep that in mind, I run through glycolysis. And in the liver, that generates ATP as well. Bless you. Citrate comes out, what do? I recycle the OAA. What have I essentially done? Look at this acetyl-CoA. Think of 2 carbons. Where'd you get the 2 carbons from? 2 of them from pyruvate. How many carbons in pyruvate? 3. I've taken one off. We'll see that in a couple of weeks. I've got 2 carbons. I activated it with CoA. The problem is I can't get it out of the liver like that. Matter of fact, I can't get fat in the liver as fat either. I've got to activate it. This CoA, I want you to note, is stuck in the matrix. It can't come out. So to get out those 2 carbons, I've got to make citrate. Out comes citrate. What do I have to do? Recycle OAA and activate the carbons again. Get them ready to fat synthesize. Here's my 2 carbon CoA. Now this in blue, I should have put it in red, flashing lights, is a key regulator. This character here, this enzyme, acetyl-CoA carboxylase, bless you, is a key regulator to start making fat. What does that mean? That means if you have lots of citrate available in the cytoplasm of the cell, this enzyme gets busy starting fat synthesis. And it makes this key intermediate, malonyl-CoA, an activated carbon. In the red dots, just for your convenience, we're seeing this circle. I want you to write next to it FAS. That's fatty acid synthase. That's an enzyme we're going to see shortly. Looks a bit like, you know, those executive toys that move the little ball from one bit to another bit, move them all around like the little magnets. That's how this enzyme works. Unlike the others, it's complex, but works in different ways. It uses an electron carrier, NADPH. It makes palmitate because it can't make anything longer. How many carbons in palmitate? Write it down. That's what you make. That palmitate then is going to be made into, up here, a triacylglycerol. What do you need? The knuckles. But you need to activate those carbons too. What do you activate them with? Phosphate, glycerol phosphate. Let's bring them all together. Fatty acyl-CoA, glycerol 3-phosphate, get 3 of them together. What have you made? A triacylglycerol. You need a driver and a cart note and delivery docket. What's your truck from the liver? VLDL. Out it goes to deliver those fats. Now, this VLDL, if you're on a high sugar, high protein diet and you're making lots of fats, where are you likely to send that VLDL to? Muscle, liver, brain, or fat cells? Which one? A, B, or C? Who picks A? Who picks B? Who picks C? The rest of them are fraidy cats. They don't know what they're talking about. Definitely to C. It's going mostly to fat. A little bit muscle, but mostly to fat cells. Any excess, you store fat. Now, this process is termed lipogenesis. You're making fat, particularly from glucose, but I want you to put underneath there also excess amino acids, i.e. those glucogenic amino acids. The ketogenic ones, they're converted to acetyl-CoA. So a ketogenic amino acid, the ones you think, oh, I'm not going to make glucose with them, are still used to make fat because they're excess to your requirements. They're carbons you're not going to use. They're not being recycled in other ways. Now, this goes on in the cytoplasm of your cell, the cytosol. Can I move to the next one? Is that a yes? Okay. Now, just like most hardcore biochemists, you have to know a few pathways. We've already seen the alanine cycle. I've hinted at the Cori cycle. We've seen the citric acid cycle. This one's called the acetyl shuttle. Why is that? Because you have a mitochondrial matrix pool of CoA and a cytoplasmic pool. The two don't transverse. What have you got to move instead? The carbons. Activate one, make your molecule, move it around to the next one. What we're seeing here is the details, let me go back one slide, of this process here. Here we've got our friend OAA, incoming glucose. There's 6 carbons down to 3, rip off one of them, make acetyl-CoA, any amino acids that are excess. What do? I make citrate, recycle the CoA. What does my citrate do? Go out through this citrate transporter. A few slides ago, I showed you the lipid bilayer with these big blue things sticking through the membrane. Integral membrane proteins. They are selective pumps and pores. This character here only lets citrate out. Nobody else in, nobody else out. A selective integral membrane protein. Where is it? The inner membrane of your matrix. It gets it out, it gets through this pore because the outer membrane's more fluid. I can get things out easier. Out here in the cytosol, what do? Grab some CoA, some ATP. What do? Recycle the acetyl-CoA, ready to make fats. What do you do with the citrate? You need this enzyme, the lyase. Who have you just made? OAA. Then another enzyme, the dehydrogenase. Some help with NADH, more electrons and protons. Malate. A name, not a structure. Malate here going to come back in through this malate alpha-ketoglutarate, write down alpha-Kg, transporter. Pyruvate also has to get in there as well. That's going in via this thing, the pyruvate transporter. So getting things in and out of membranes is critical. That's why I showed you that image a while ago, those blue structures that are integral membrane proteins. Selective channels and pores, some of them pumps, some of them enzymes. Indeed, ATP synthase is an enzyme stuck in the membrane. We done with this one? Okay, you need to know the acetyl shuttle. What's it for? For getting carbons out of the mitochondrial matrix into the cytosol to do what? Make fat. The question I want to pose to you, what if you suddenly don't have a high-fat diet and you need more blood glucose? At the instant, we have our citrate back out. Do you think these hepatocytes would keep making fat or start potentially making sugar, making glucose? What do you think? Of course, your liver has to respond to that on a second-by-second basis. So glucose is high, make fat. When glucose drops, stop making fat. Come back to making glucose. Let's move to the next one, shall we? Okay. To do that, you need some help. And the help you need is this character, biotin. Some of you have heard of biotin. If you do any biotechnology, we use biotin with avidin. They stick together better than Velcro. We use them to find things within cells. This conversion of acetyl-CoA to malonyl-CoA, where's that? Let's go back here. See this acetyl-CoA in the cytosol to malonyl-CoA, this key regulatory enzyme? That's this step here. Acetyl-CoA, there's my carbons, there's my CoA. What do I need? Biotin. Biotin's going to grab some CO2, not the oxygen, but the carbon. What's it going to make with the help of ATP? Malonyl-CoA. Notice the dashed line there is the CO2 component that you've just used. Where are you? The mitochondrial matrix component. You've come out, you're in the cytosol of what? A liver cell. Lots of CO2 there. So you don't actually exhale all the CO2 you manufacture. But like a lot of things we're going to see during the semester, this particular pathway is unidirectional. That's what it says here. This is irreversible. Once I make malonyl-CoA, so if I'm on a high sugar diet and this acetyl-CoA doesn't go back in, I make malonyl-CoA, I'm committed to making fat. You clear on that? So there's your liver. What's it doing right now? It's making fat, isn't it? Through malonyl-CoA. Can I move to the next one? Yes? We're right to go? Cool. Biotin. Now I want you to think of biotin, it's kind of like an enzyme with two heads. Biotin grabs CO2 and swings from one active site to another active site, which is we're getting ready for the FAS enzyme. Remember I said FAS was a bit like an executive toy. The ball moves from magnet to magnet to magnet, which is each active site. Here's the first step, giving an idea of the concept. This enzyme has two active sites. I grab CO2 on one, start the reaction. I then move it across on our lipo arm to the other active site. I haven't lost that carbon. Then I produce my product. Go back and start again. That's what we're seeing here. We're seeing this long chain carbon with this thing, the biotin arm, some NH functions here. That HCO3 is the carbon we're seeing in the CO2. In comes the ATP to help us. Our carboxylase, whatever you put, carbon on that nitrogen. You put it in one site and move it to the other site. What have you produced? Malonyl-CoA. You look down below, you can see that blue part is one hand. This white part is the other hand. This lipo arm, which is a long chain carbon to allow it to flex from one active site to another, is simply grabbing carbon, moving it to another site to make malonyl-CoA. Are we clear with that concept? Two active sites to make one product. One active site grabs carbon, the other active site makes the product. This is important for biosynthesis and gluconeogenesis. Can I move to the next one? Yeah, so if you look here, you've got one active site grabbing the carbon. Here is CO2. That arm swings around to the other active site and produce your product, which is malonyl-CoA. Exactly. So that arm grabs the carbon, swings it across the other side. Carbon product, it's empty, it goes back to the other arm, grabs another carbon. So it keeps on doing it, one side to the other, which is important for this enzyme. It's not enzyme channeling. That's more like a production line. Product goes to another enzyme. This is the one complex protein, two active sites. Can I move to the next one? The big exam questions on this, guaranteed. Okay, let's start making fat, shall we? This character here, fatty acid synthase, what's the acronym you're going to use? FAS. It has 7 catalytic sites, not just 2 hands, it has 7. 2 plus another 5 active sites. Think of that executive toy, 7 places for that ball to go to for something to happen. How does it swing it around? If you look in this red box here, we have a long chain carbon called a pantothenic acid. Didn't we see that a little while ago with something like CoA? What have we got here? This thing is linked to this protein. This ACP is the acyl carrier protein. Down the end of it, I want you to circle that sulfhydryl. What does the sulfhydryl tell you? Somewhere I can activate somebody. It's kind of like a small, weaker version of phosphate. Who's going to grab that? Well, I need to go back 1 slide, or 2, or 3, or 4. Citrate, acetyl-CoA, malonyl-CoA. Yeah? I'm about to go into fat synthesis on this big complex protein with 7 catalytic sites. Write it on here now. FAS, 7 catalytic sites. Oops. I'm just going to grab malonyl-CoA. I'm going to use the sulfur to do the work. But I'm then going to swing around from active site to active site. Can we go to the next slide? Not yet. Now, fortunately, I have an animation for this particular activity because it's kind of complex to think about. But you've all seen those executive toys. They're not the ones that just hit the ball one side to the other, but the little magnet that can kind of swing from one part to another depending on how you push it. So I want you to think like that. There is my particular carbons coming in. I move from active site to active site. Can I move on now? Yes? Anybody else right? Okay. Forget the schematic for a moment. Let's watch the video. He says with crossed fingers. Okay. Last slide, you saw the acyl carrier protein. There's a little ball. Here it is here. You see it up here, a little protein component stuck to this kind of 2 or Z-shaped protein. Each of those coloured regions is an active enzyme site. This is one long polypeptide, one big protein with different active sites, not just 2 now, but 7. It has this little arm, the string that's hanging from the cable. We're going to grab protein and move it around to active site. So this is your ACP. I'll put it around a blue box there. What's going to come in? There's your free sulfhydryl. Here comes malonyl-CoA. Here's the animation. Now this animation will be up on views for you, like they all will be as we go through the semester. First I take you through them and you can watch them as much as you want. So here comes my malonyl-CoA. Oh, hang on a sec, that's not malonyl-CoA. That's acetyl-CoA. What the hell's going on? We need to prime the system. We're going to prime this sulfhydryl acyl carrier protein with 2 carbons for a start. Remember, this ACP swings around. We've just swung it around and we're going to move the 2 carbons to this ACP synthase site, which is one of these. You see it just here. So I've grabbed 2 carbons on a CoA and then slipped them away. Now I'm ready to go. I've activated my enzyme, my FAS. Now I need malonyl-CoA. Incoming. Heads down, helmets on. Here comes my malonyl-CoA. I'm hooked up to ACP now. That's a different active site. What do I recycle? The CoA. I've still got a sulfur. So I've got a sulfur attached to my ACP, this character up here. I've now got my malonyl-CoA. There's the carbon that I grabbed from CO2 in biotin just a little while ago. And this is complex, isn't it? I've activated the enzyme with acetyl, but I've got to bring these 2 carbons back. Bring them back, add them to the chain. What have I just done? Almost. There we go. The 2-carbon acetyl that was over here is now here. What did I do with that extra CO2 bit that came on malonyl? Out goes the CO2 again. So poor old biotin did the work to grab CO2, stick it on malonyl-CoA. This character said, up the CO2, off you go. All I'm interested in is the 2 carbons. Now I want you to think about this for a moment. Glucose has 6. You split it into half to make 3. Pyruvate's 3, but all the work's done on 2 carbons. 2 to add, 2 to subtract. Think of even addition and even subtraction. The 3 carbons kind of moves around the cell. Out comes CO2. What do you do with that? You might recycle it on biotin, but you might also exhale it. So if you're having lots of fat in your diet, what are you potentially exhaling? A lot more CO2. A bit like a cow, aren't you? Not methane, it's CO2 you're producing. But you're currently making fat, producing CO2. Let's keep going and see what happens next. Remember, the executive toy has to move from site to site. In comes some protons. What are you going to do? Now remember, you're making fat. Let's go back a tad. What have you got there? Ketone. I have to remove the ketone. I have to remove anything and just produce fat. Your cells produce saturated 16-carbon fat. I've got to get rid of the oxygen and the hydrogen. Let's do that in each of these active sites. You see this thing swinging around? So I grab some protons. I've got an alcohol there I have to get rid of. I've got a double bond. I need more protons, don't I? Different active site. The site's a bit slower. More protons. Here they come. NADPH. What does your liver cell do? Makes lots of NADPH. I've just now made a 4-carbon fat. Now, what do you think might be the next step? I need malonyl-CoA. What should I do? Get these characters out of the way. Bring in malonyl-CoA. Same sequence as before. Bring them back, ditch CO2, get rid of the double bond oxygen, add protons as hydrogen, get rid of the double bonds, keep on going, get these characters out of the way, bring in another malonyl-CoA. Kind of comfortable with that sequence? You can see the 2 carbons addition at a time. The 3 carbon, the third one, gets ditched out as CO2. But every time I have an addition, I have to move the chain out of the way, bring in malonyl-CoA, bring it back on. Yeah? And that is why you only make a 16-carbon fat, because it gets so long, that's as many carbons as this enzyme can handle, then it becomes sterically unstable. Enzyme says, I can't take anything longer than that, that's all I'm going to make. I also want you to jot down, it doesn't make double bonds. You're not producing double bonds in this fat. So the fat you make is more like beef fat. What did you have to do? You had to prime it. That's why you have the acetyl-CoA, and each one of these is an incoming malonyl, 3 carbons, ditched out 1, off you go. You need to know the key steps in this pathway. Activation, get out the way. Malonyl-CoA, bring it back. Get rid of the CO2, get rid of all the ketones, add protons. Make a saturated fat. Get the 16 carbons, off you go, next one. Comfortable with those steps? Now I've made a 16 carbon fat. Let me go back a couple of steps here. I've made a 16 carbon fat. What do I have to do? Activate it, put it on glycerol, make TAG, send it out into VLDLs. What's going on in your liver right now? All of this and VLDLs circulating around your blood, trying to deliver fat to where? Adipocytes, fat cells. You need to know this for the exam. Always exam questions on fat synthesis. Now, if you think about fat oxidation, I said beta oxidation. What's that? The 2nd carbon. Next week we'll see that, beta oxidation. What do you generate? The reverse of this, electrons and protons. Now, other species can go longer. Some other species can add double bonds. You cannot. That's why you have to ingest those other species to get the double bonds to change what's wrapped around your neuronal cells to make them more functional, or make those second messengers like arachidonate. You can't make those so easily. You have to ingest them. Can I move to the next one? Anybody else right? This is summarising it for you. Why would I summarise it? Because it's probably important. There you see the addition of the carbons all the way along from your ACP protein on each of the active sites. The bottom line is this equation. Now, I don't ask for many equations in this unit, but this one you might find bites you in the exam, maybe on your gluteus maximus. 8 acetyl, what's that? 8 activated 2 carbons. 8 times 2 gives me what? 16. 14 NADPHs, 14 protons, 7 ATPs. What does that tell me? Making fat is a cost to your liver cell. If that liver cell runs out of ATP, it's got lots of AMP, it doesn't make fat. It stops the process. There's no ATP for it to make fat. It goes back and makes ATP. What's your product? Palmitate. Right under it, C16. Delta 0. No double bonds. What have you done? Recycled the CoA, 14 NAD pluses because you've got rid of the hydrogens, 7 ADPs, lots of inorganic phosphate, and you've just become a desalination plant. You've made water as well. Not there yet. Ready to move on? Anybody else right? 1 second. 1 second's up. Let's move on. All set? Trying to type in that equation. Might take a little while. Hand drawing's not a bad thing, but not in your team leader summary slide. Don't do hand drawn stuff. I'll move on now. Cool. Let's talk a little bit about the regulation. When you make fat, when you don't make fat. Well, I said if you have lots of glucose, you're going to make fat. When do you have lots of glucose? After a meal. What happens after a meal? Doesn't your blood insulin go up? Trying to put glucose into storage in cells. That's what it's trying to do. It's going to activate, and that's what the little plus is, this enzyme, a phosphatase. And you know what a phosphatase does, don't you? You told me. Somebody's going to have their phosphate ripped off. Who is that phosphate? Acetyl-CoA carboxylase phosphate. There is an enzyme with a phosphate on it that's inactive. Insulin activates an enzyme to activate the removal of the phosphate to make acetyl-CoA carboxylase. What does that character do? Isn't that kind of important to make malonyl-CoA? Let's think about that for a moment. Just had a big meal of lots of fat and lots of sugar, mostly lots of sugar. What happens? My blood insulin went up. I'm telling cells to put more glucose transporters, suck in the glucose. At the same time, I'm telling an enzyme to activate an enzyme to do what? Make fat. That's what you're seeing here. This enzyme's now busy. Wasn't this a key regulator? There it is busy turning this acetyl-CoA into malonyl-CoA. You'll see there's a little bit of reversibility. Why is that? Because I said when you commit to it, you have to keep on going past the malonyl. But until then, my blood glucose drops, I might need those carbons to make glucose in the liver. If I'm making lots of fat and making lots of palmitoyl-CoA, let's go back here. Where is he? There he is. I'm making lots of this. There's my lots of fat. And I'm not sending out VLDLs. What am I telling my cells? It's not setting off, not sending the trucks out of Melbourne. So stop making fat because the trucks can't leave Melbourne. That's what you're doing. You're inhibiting the process. Back here. If that builds up in the cell, put a square bracket around it, arrow up, in the cell. It's not going out. It feeds back to this carboxylase and says, slow down. But you also see these characters, glucagon and adrenaline. Now my liver cell's busy making fat, and suddenly I have to do a metabolism quiz, and what happens to your heart rate? Totally. I walk in here, what happens to your heart rate? It skyrockets. What do? Dump out glucose. What do you tell your liver to do? Stop the hell making fat and start making glucose. What are you going to do? Inactivate this enzyme. You're going to use a phosphatase, a kinase, I should say, to put onto this enzyme and inactivate it. Isn't glucagon kind of the inverse of insulin? Insulin, lots of glucose, make fat. Glucagon, not much glucose, don't make fat. Kind of clear on that? So you keep seesawing hour by hour, minute by minute, depending on what's going on and what your cells are doing. Done with this one? Is that right? Yes? Okay. Oh, what do you know? Your heart rate's up. What are you doing with your fat synthesis right now? Are we ready yet? I like a solid no. Definitive response. None of this namby-pamby no stuff. Maybe. Are we there yet? Somebody is. Sorry? Good job, LPL, go for it. All caps, LPL. Why would I say LPL? So you remember it. Just before you go, just before you go, just keep in mind those report part A topics will be on views, 1 o'clock on Thursday in the report folder. Okay? You might want to look at them before next week.
Is there a difference between synthase and synthetase? Is that a type? There is a difference between synthase.
I had a last question about that, about one last slide. Now, I saw that you ATP and ADP.
Use ATP to inactivate it.
Oh, so that's to inactivate. I thought it was to activate.
Yeah, so you use a kinase, grab a phosphate, and put a phosphate onto the enzyme.
That's to inactivate. And then the ATP activates.
The phosphatase takes the phosphate off.
And that just activates. So what is it? So the ATP doesn't activate it.
Or inactive. Yeah.
So
So the ATP, the phosphate on there, see that phosphate? That goes in there, that's inactive. Take that phosphate off. Now remember I said before that sometimes you see enzymes that are activated with phosphate. This is a reverse. You're on now?